Okay, so today's lecture is on sex differences in skeletal muscle metabolism. First off, and as always, a couple of announcements. First, Tuesday, February 11th, tomorrow at 5 o'clock p.m. in Pond Inlet is Brock's first International Day for Women and Girls in Science. This is an opportunity to come out and support female faculty as well as female graduate students as they talk about some of their current research endeavors. Appetite hormone papers have been posted to Sakai, so if you're presenting on that topic, go ahead and get started. Case study number 2 should be posted by the end of the week. Thank you everyone for submitting case study number 1. Paper summaries will be also posted by the end of the week, so those reviews of the papers that have been presented on so far will be posted in an orderly way for you guys to study for the midterm. The midterm is Thursday, February 27th in class, so that's 2.5 weeks from now. So important that you guys are starting to think about it. For the midterm review, we discussed in class this idea that you guys are to email me questions over reading week, and I'll put together a PowerPoint presentation based off of your questions so that we can discuss them on that day. If you guys show up to the midterm review, odds are that I will probably give you guys some hints as well as maybe one or two of the multiple choice questions. So today's lecture, we'll have an introduction to sex differences, then we'll talk about how these sex differences relate to both lipid and carbohydrate metabolism before we shift gears and think about protein metabolism. So quick introduction. It's clear that sex differences in human health or even rodent health, biology, nutritional sciences, there's this pressing issue where there is a sex gap, meaning that women are underrepresented in this area of research. And this is seen by a number of titles I've pulled here from recent publications saying, "Where are women in sports science research? The gap has not been resolved since 1983. Exercise in sports science is failing by design in understanding female athletes." And it's really all encompassing this idea that in 2020, sex differences are really at the forefront of research, and it's something that researchers are starting to consider. First, I want to give a historical perspective on where we came from in relation to studying and understanding sex differences and why this gap still exists today. So in 1993, the National Institute of Health, which is a US organization, mandated the inclusion of female research participants in health science research. This was termed the Revitalization Act. And under this Revitalization Act, any research that was conducted in the health sciences, both men and women had to be included, unless there was explicit reasons why women did not belong in this area of research, for example, things like prostate cancer research. But really, this was a slow and arduous process. It wasn't overnight that women were really started to be included, and it wasn't really until the year 2000 that researchers began to acknowledge that women are not simply small men in terms of their physiology and metabolism, meaning that researchers began to accept that true physiological and metabolic differences might exist between the sexes and that it's important to tease apart what these differences are and why they exist. With that being said, so only since 2000, it's only been 20 years, this is still an area of research that is relatively untapped, and only recently, in the last 3 to 5 years, has there been a new push to study sex differences in metabolism. Another thing I want to touch on is that National Institute of Health mandating the inclusion of female participants under the Revitalization Act. This is a policy that's only been in place in the United States. No other country has this kind of policy. And we think about the big players in biological research, whether that's Canada, Scandinavian countries, they don't have these same inclusion policies as the United States does, which contributes to this idea that, again, it's a slow and arduous process for the inclusion of female participants as research subjects. When we consider sex hormones in terms of their role in physiology and metabolism, female sex hormones, we're considering estrogens and progestogens. Estrogens are broken down into 3 main estrogens: estradiol, estrone, and estriol, where estradiol is the most potent and most abundant estrogen that's found in circulation. And this is going to be the type of estrogen that you typically see used in a research setting, whether it's delivered to humans or rodents. When we talk about progestogens, the most common type that we're talking about is progesterone, where most other progestogens are actually just metabolites of progesterone. On the flip side, when we consider male sex hormones, we're thinking about androgens. The key androgen that we're thinking about is testosterone. And aromatase is, importantly, an enzyme that we can actually use to convert testosterone to estrogen within males, and this may have implications in one of the studies discussed at the end of this lecture in the protein metabolism section. So when we go on to think about a typical menstrual cycle, because this is where we're going to see fluctuations in those female sex hormones, what we typically think of is this menstrual cycle that's about 28 days on average. What we see is that day 1 is marked as menses, or the onset of bleeding. This is when estrogen and progesterone levels are both low. And if we follow the orange curve here, we see that estrogen levels rise throughout the menstrual cycle, peaking just prior to ovulation, dropping off in the back half before they rise again around day 21. When we think about progesterone, we're thinking about relatively stable, low levels throughout the first half of that menstrual cycle. Around ovulation, just following ovulation, we actually see this rapid rise in progesterone levels that drop off around the 28th day of that cycle. Importantly, ovulation is on day 14 of that menstrual cycle. And what we can actually do is divide these menstrual cycle phases into 4 key phases, whether that's the early follicular phase marked by days 1 to 7. This is typically defined as low estrogen, low progesterone levels. The late follicular phase, where estrogen levels are elevated as they peak in this phase. The early luteal phase, when progesterone levels begin to rise and peak by day 21. And the late luteal phase, where progesterone levels begin to decline back to their baseline levels as we prepare for the next stage of menses. This is going to be important to consider when we think about the recruitment of female research participants. So typically what you'll see in a research study is participants are recruited in the follicular phase, meaning that there's low estrogen, low progesterone levels. They might be recruited at ovulation, so high estrogen, low progesterone, or they might be recruited in the luteal phase when both estrogen and progesterone are elevated above baseline, and really when those peak progesterone levels are seen. The problem here resides in the fact that, one, there's not a lot of data with female research participants, and the data that does exist uses a mixture of participants in the follicular phase, some at ovulation, and some at the luteal phase. So when we try to compare the research across studies, it's very difficult because these different hormonal profiles make it difficult to interpret what's actually happening or how these different hormones may be influencing, in our case, skeletal muscle metabolism. Moreover, what's important to consider when we're recruiting female research participants is that we want to use the appropriate menstrual cycle phase that addresses our research question. So for example, if we were considering the effects of estrogen on a specific skeletal muscle outcome, it'd be important for us to test in both a high and a low estrus phase. So we'd want to have, in particular, the follicular phase as well as ovulation, as this would tell us differences in how estrogen may be influencing our skeletal muscle metabolism. How do we actually go ahead and confirm menstrual cycle phase? So as good scientists, it's important that we're able to report and show that our participants were actually in a specific menstrual cycle phase, and that this coincides with obviously the phase of interest that we're looking at. First is the calendar method, so literally just tracking the onset of menses, the length of the cycle. We're able to determine roughly, basing off the menstrual cycle length, which phase the participant might be in, whether that is the follicular phase, luteal phase, early or late. We can have an educated guess using this method. We can also use basal body temperature. So what we know is that core temperature rises about 0.3 to 0.5 degrees Celsius during the luteal phase of the menstrual cycle. So we can now tease apart follicular versus luteal phase based off of core temperature changes. We can also look at urinary luteinizing hormone analysis. So what's important to note here is that this is a dipstick test. You take a small urine sample, you put this piece of almost paper-looking thing inside the urine, take it out, and what you can see is that luteinizing hormone, which coincides with estrogen levels and the peak in estrogen, will actually be measured or confirmed using this dipstick test. So this is important analytic technique for confirmation of ovulation. Of course, you guys know that we can use blood and salivary markers to determine estrogen progesterone levels. We can also use a technique called serial follicular scanning. What this does is we can actually take an ultrasound to measure the size and number of the follicles present. And this, based off of the size and the number, we can actually correlate this to different menstrual cycle phases. This is kind of the gold standard for this field. But of course, due to lack of equipment or the cost of using this, as well as the invasiveness of this procedure, a lot of people would not opt for this methodology. And the most common ones that you'll see would probably be basal body temperature, as well as blood and salivary markers. So one thing I want to touch on before moving into some of the literature was this idea of oral contraceptive use. So what I want to highlight is that a lot of the literature, or all of the literature that we're discussing today, does not involve oral contraceptive users, and that's because of a lot of complexity associated with oral contraceptive use. But importantly, oral contraceptives are the most common form of hormonal contraception. There's a statistic out there that about 80% of Canadian women will use oral contraceptives at some point in their reproductive life. And oral contraceptives have existed since the 1960s. What oral contraceptives are is synthetic sex hormones, whether that is ethinyl estradiol in combination with progestin or progestin only. But again, as I mentioned, there's lots of ambiguity with relation to oral contraceptive use and how that might link to human physiology and skeletal muscle metabolism. Just to name a few, we can have oral contraceptives that are a combined pill, meaning they deliver ethinyl estradiol as well as progestin, or they can be progestin only. The pill can be monophasic, meaning that all of the progestin concentrations in the pills is the same. It can be biphasic, meaning that for half of the cycle, the progestin concentration is one dose, and for the other half, it's another dose. Or it can be triphasic, meaning that the progestin concentration changes every week that you're taking the pill. Moreover, the generation or the formulation of these oral contraceptives can be either first, second, third, or fourth generation. This just means that in all oral contraceptives, ethinyl estradiol is the synthetic estrogen we use, and the progestin can be first, second, third, or fourth generation. This is based off of the pharmacokinetics as well as the pharmacology behind these different oral contraceptives. And really, what the goal was is that we want to reduce the side effects with each progressive generation of these oral contraceptives, but it also means that the way they're metabolized, the way that they're broken down, the chemical properties associated are all different. And of course, if you surveyed a room full of women taking oral contraceptives, there would be a very big scatter between those on first, second, third, or fourth generation oral contraceptives, making it even more difficult when we consider these individuals for research purposes, because it adds more complexity, more ambiguity to our findings. Lastly, when we consider pharmacokinetic profiles, there's not much work out there that shows us how these oral contraceptives are metabolized, whether they are broken down by first pass metabolism of the liver, how much is absorbed, how quickly. Again, this would all differ with women taking different brands, different types, different pharmacologically produced oral contraceptives. And really, big picture, we don't really know. We have no idea how these different oral contraceptives and their formulations influence skeletal muscle metabolism. So for this reason, we're not going to be talking about studies within oral contraceptives for this class. One, because the research isn't as well backed, and two, because not as much research exists in this realm. So first I want to talk about some whole body differences that are important to touch on with regards to sex differences. But just note that this won't be the focus of today's lecture, but I think they're important issues to address before we delve in and focus and hone on skeletal muscle. So first and foremost, body composition. Women predominantly accumulate subcutaneous adipose tissue, meaning this is the adipose tissue or the fat that we're able to pinch between our fingers, whereas males accumulate visceral adipose tissue. This is the fat that is around our internal organs and is able to act as that kind of shock absorber, but also the type of adipose tissue that is predominantly linked to metabolic disease or poor health outcomes. Interestingly, when women enter a menopausal state, so decline in estrogen, progesterone, or female sex hormone levels, they're actually similar to men in that they gain more visceral adipose tissue. This suggests that estrogen maybe plays this protective role and deposits adipose tissue subcutaneously where it's more safe, as opposed to viscerally where it plays important roles in dictating metabolic health. Moreover, females have greater fat mass and lower lean mass compared to their male counterparts. So for comparison, males are typically 10 to 15% fat mass, whereas females are typically 20 to 30% of their body mass as fat mass. When we go on to talk about lean mass, we're thinking about skeletal muscle properties. So females have a greater proportion of type 1 muscle fibers compared to males. And accordingly, since this is proportional, males have a greater proportion of type 2 muscle fibers. So females more oxidative, males more glycolytic. Moreover, males have muscles with greater cross-sectional area compared to females. So this is relating big picture to this idea that males have more lean mass. So this table on the bottom here is pulled from a 2019 paper, just showing you the relative percentage of muscle fiber types from the vastus lateralis in males and females. So for example, type 1 oxidative fibers, females are 64%, whereas males are 59%. Again, females are more oxidative than males. And if we combine the type 2 muscle fibers, we see males are significantly more glycolytic than females. It's important to note that they use the vastus lateralis in this example because the vastus lateralis, again, is about 50% oxidative, 50% glycolytic. So this would be a good place to see these muscle fiber type differences between the sexes. Lastly, I just want to talk about oxygen transport. So in the context of breathing, we take in oxygen. That oxygen is going to be bound to hemoglobin, delivered to, in our case, the skeletal muscle, offloaded. That oxygen can be used for aerobic energy provision. What we know is that women actually have lower mean hemoglobin levels. This is approximately 12% lower than men. Not only that, females have lower cardiac output. And remember, cardiac output is a product of heart rate and stroke volume. And cardiac output is actually lower in females when expressed both absolute and relative to body size. The reason why this exists, or why this phenomenon is a thing, and what we discussed in class, is the idea that females not only have smaller hearts, but a decreased capacity to actually use the heart to pump blood. So stroke volume is the volume of blood ejected per each beat of the heart. And women have a reduced stroke volume, meaning a reduced cardiac output, because maximum heart rate, or heart rate in general, is basically the same between the sexes. So big picture, women have a decreased capacity for oxygen transport compared to their male counterparts, which may have an implication for fatigability, for skeletal muscle metabolism. and really how we use oxygen for aerobic metabolism. So now I just want to take a step back, set the stage in terms of what we're talking about in terms of substrate utilization. Just a refresher for you guys, make sure we're all on the same page. So when we think of the classic fuel utilization charts here, remember, we're thinking that during exercise, we rely on both carbohydrates and fats for fuel. These carbohydrates and fats are found both in the plasma, meaning in the circulation, so blood, as well as within the skeletal muscle itself. If we look at the figure on the top, we're thinking about exercise intensity. So VO2 max on the x-axis, fuel substrate utilization along the y. What we see is that at low exercise intensities, free fatty acids or plasma free fatty acids predominate as a fuel source. And as exercise intensity increases, our reliance on skeletal muscle glycogen and glucose increases significantly. Moreover, when we turn our direction to the bottom figure, looking at duration of exercise, what we see is that as that duration of exercise increases, we have a heavier reliance on plasma-free fatty acids for fuel. So now we're going to direct our attention to sex differences and how they relate to lipid metabolism. So this red box here just showing you to ignore the glucose glycolytic side of things, really focusing on the lipid side of the picture. So what we do is we take lipids, liberate them into their individual free fatty acids in the circulation. Those free fatty acids are brought across the plasma membrane through fat transporters. So fat CD36 is a key fat transporter. Those free fatty acids can then be turned to fatty acyl-CoA and then to fatty acyl-carnitine via CPT1. Fatty acyl-carnitine, we know it can cross the outer and inner mitochondrial membranes. CPT2 converts it back into long-chain fatty acyl-CoA. Beta oxidation can occur, Krebs cycle can occur, electron transport chain can occur, energy provision through fat metabolism is our goal. Alternatively, in the bold red letters looking at intramuscular triglycerides, we can also liberate free fatty acids from those stores as well, and they proceed through the same pathways. So what I want to talk about first is big picture, so whole body measurements of substrate utilization and how they might differ between the sexes before we actually delve into the enzymes, the transporters, or different metabolic machinery that we might have and where those differences might reside. So first thing we'll consider is substrate utilization at rest. So remember, RER, or the respiratory exchange ratio, is defined as VCO2 over VO2. RER can show us the relative contribution of carbohydrates and fat for fuel at rest and during exercise, where an RER of 0.7 dictates fat utilization and an RER of 1 dictates carbohydrate utilization. What the literature shows, and highlighted in these red boxes, is that there's no sex difference between men and women in fuel utilization at rest. So that first top figure, table 3, is looking at resting metabolic rate. So if we laid still on a table, took oxygen consumption and carbon dioxide production measurements, there would be no differences between men and women. So 0.8 versus 0.79, same RER. And the other study took resting measurement prior to exercise. Again, 0.74 versus 0.71. No difference, no significant difference in fuel utilization between the sexes with regards to resting substrate utilization. However, when we divert our attention to substrate utilization during exercise, again, an RER of 0.7 represents 100% fat utilization, and an RER 1.0 represents 100% carbohydrate utilization. What we see is that during submaximal exercise, females rely more heavily on fat for fuel, or lipids for fuel. This can be depicted on the graph, looking at RER with the highlighting of 0.87 as the RER for females in the black circles, and an RER of 0.93 in the white open circles for males. So same exercise intensity, same exercise duration, females rely more on fat for fuel. And this is shown in table 3 here when we see calculated fuel consumption. If we actually translate those RERs into tangible numbers, so the grams of lipid utilized or the grams of carbohydrate utilized, what we see is that females use more grams of lipid for the same exercise bout and fewer grams of carbohydrate, whereas males use less grams of lipid and more grams of carbohydrate to fuel that exercise bout. So how I like to think of this fuel utilization paradigm is this idea of a car. You would never see a difference in how a car uses gas or uses fuel at rest when it's idling in a driveway. However, these differences become apparent when you are driving the car down the highway, down the QEW, and what we see is that with these different styles of driving, or more testing the system, we can actually see differences in how that fuel is used, and we can see sex differences in how substrate is used for exercise. So now we need to think about so we know the divergent difference between men and women as adults is their sex hormone profiles. So we need to think about which sex hormone is the driving factor for these differences. So what this study did was recruited 9 healthy, active men. Fairly invasive study because what they did was they performed an acute bout of exercise in 3 physiological conditions. The first, physiological testosterone levels. This is a control condition, no manipulation in place. 2, they did a low testosterone condition. So what they did was they actually have a drug that can pharmacologically suppress testosterone production, and they did this for about 3 to 5 days. They also had a third condition where they had high testosterone levels, where they actually put a patch on their arm and gave them transdermal, so across the skin, delivery of testosterone. What they did in each of these 3 conditions was they actually tested submaximal exercise intensities, and what they found when looking at substrate utilization during these submaximal exercise intensities was that testosterone levels, whether physiological, low, or high, as indicated by the 3 bars, there was no difference in fuel utilization with those different testosterone levels. So the amount of carb, the amount of fat did not change despite changes in testosterone levels. This led the researchers and future researchers to really question then, if testosterone isn't the driving factor, is it female sex hormones, predominantly estrogen, that is the driving factor? So that's when this study came along 15 years later, where they had 12 healthy active men. And again, what they did was a submaximal bout of exercise, 90 minutes at 65% VO2 peak. And they had 2 conditions. The men either cycled in a control group, so no intervention, for 8 days, tested them on the 8th day, or they supplemented with exogenous estrogen for 8 days. Following the 8th day, they then performed the same exercise bout. What they found, looking at the figure on the side here, is that in the black bars, or the black squares, being estrogen, and the white squares being placebo, estrogen supplementation acutely for 8 days significantly decreased RER, meaning estrogen supplementation significantly reduced carbohydrate oxidation, about 5 to 16%, and it significantly increased lipid oxidation, or fat utilization, 22 to 44%. So this is a pretty strong indicator that even an acute supplementation of men with estrogen can have dramatic influences on substrate metabolism, leading us to believe estrogen may be a key influential factor driving these differences between the sexes. So really this leads us to question how? What is the mechanism of action? How does estrogen elicit these effects on the skeletal muscle metabolism? So here we have the estrogen signaling cascade, and what we know is that estrogen is a fat-soluble molecule, meaning because it's a steroid hormone, it's fat-soluble, it can freely diffuse across the plasma membrane. Once it crosses the plasma membrane, it binds to an estrogen receptor in the cytosol, which is ERC. It can then enter the nucleus, where it binds to something called an estrogen response element, or ERE. When bound to that estrogen response element, it can then go on to influence, either decrease or increase, mRNA through transcription, and then go on to influence protein synthesis through translation. How this actually ties to carbohydrate and fat metabolism is we might have upregulation of genes, so mRNA, as well as protein content related to lipid metabolism, whereas we might have different influences of estrogen on mRNA and protein content related to carbohydrate metabolism, whether that is enzymes or transporters or other metabolic machinery. So again here, just the red square, meaning we're not focusing on carbohydrate yet. We're really focusing on, again, these specific pathways related to lipid metabolism. And now we're going to delve into the individual enzymes, the individual transporters, and how they might actually differ or not between the sexes. So the first thing I want to talk about is lipoprotein lipase. So here we're focusing on taking triglycerides, liberating free fatty acids within the circulation, so in the blood. Importantly, we talked about in class this idea that, not to confuse you, but lipoprotein lipase is another lipase like hormone-sensitive lipase, or HSL, and adipose triglyceride lipase, ATGL, meaning that its role is to liberate free fatty acids from triglycerides in order to, again, provide those free fatty acids to be uptaken into skeletal muscle and used for fuel. So when we're talking about now moving from taking triglycerides, liberating those free fatty acids into the circulation, this study looked at 22 females, 24 males, different training statuses. So NT means sedentary or not trained, recreationally active, MT or moderately trained, endurance trained would be ET. What we see looking at the first figure here is the mRNA expression of lipoprotein lipase. And we see that in females, lipoprotein lipase mRNA is higher than their male counterparts, regardless of training status. When we go on to actually express this as protein content, which is not shown, or activity, we actually see that mRNA does not translate to increases in protein content or activity. And again, highlighting back to the idea as discussed in some of the presentation sections, we talked about this idea that mRNA does not always translate to protein content, does not always translate to activity of an enzyme. So here's one of those prime examples, elevated mRNA for lipoprotein lipase in females, but we don't see that translate to an increase in lipoprotein lipase protein content or activity. So what we actually see here is that it appears to be a better link between lipoprotein lipase activity and training status. So we see moderately trained or endurance trained individuals, whether they're male or female, have higher lipoprotein lipase activity compared to those that are sedentary or not trained. Again, no sex difference in relation to lipoprotein lipase activity. But when we actually look at circulating plasma free fatty acids, what we see is that despite no difference in lipoprotein lipase activity, remember, LPL takes triglycerides, liberates free fatty acids in the circulation, we still see this rise in plasma free fatty acids during submaximal exercise in females compared to males. So looking at this figure here, we're looking at rest as well as 90 minutes of submaximal exercise, females in the black circles, males in the open white circles. What we see is that plasma free fatty acids are higher, so circulating free fatty acids are higher in females compared to males. The mechanism behind this is not 100% clear, but regardless, we now need to consider is that females have these higher circulating plasma free fatty acids. How do we accommodate for this? Is there an increase in fatty acid transporters at the plasma membrane that can increase our fat transport into skeletal muscle for use for oxidation and ATP provision? So what we see is that when looking at plasma membrane lipid transport, fat CD36 protein content is significantly higher in females compared to males, regardless of, again, training status. So not trained, moderately trained, endurance trained. Females, it's always higher. Taken together, this suggests an increased capacity to transport fatty acids into skeletal muscle in females compared to males. So now that we've talked about taking in free fatty acids from the circulation across that plasma membrane, we also need to think about this idea that we can actually liberate free fatty acids from intramuscular stores. So is there a difference in how we use intramuscular triglycerides for fuel or for free fatty acids during exercise? Reminder, intramuscular triglycerides are important as a fuel source during exercise because they're in close proximity to the mitochondria, meaning they can rapidly provide an energy source for fuel provision. So what the study did was looked at intramuscular triglyceride levels in both males and females, 5 males, 7 females. What they found was that 7 weeks of endurance exercise training resulted in, basically what they did was they took resting muscle biopsies both prior to this training protocol and after this training protocol, but 7 weeks of endurance exercise training resulted in significant increases in the number of lipid droplets, so the number of IMCL. So IMCL is the same thing as IMTG, just for your own notes. And what they also noticed was that regardless of training status, females had higher or a greater number of lipid droplets compared to males, meaning that females have a greater capacity for intramuscular triglyceride storage. But what we really need to think about now is there a difference in intramuscular triglyceride utilization between the sexes? So when we think about the ability to mobilize intramuscular triglycerides, this study comes to mind. Similar findings as the previous studies. So females have these greater intramuscular triglyceride stores. So women compared to men at rest here, women in the black bars, men in the white. Higher intramuscular triglyceride levels in the females. And what we see is that when we look at the capacity to utilize these intramuscular triglycerides, we actually see that females who have higher intramuscular triglyceride concentrations, MCTG also means IMTG, have a greater capacity to utilize those intramuscular triglyceride stores. So we see this positive linear relationship. So women are these black dots, men are the white dots here. We see higher intramuscular triglyceride concentrations, higher IMTG utilization during exercise. How much? Approximately 25% greater utilization of intramuscular triglycerides for fuel in females compared to males. So this leads us to question, how does this actually happen? What's the mechanism driving this? We know females have greater intramuscular triglyceride stores. We know they can use them to a greater extent during exercise, but how does this happen? So this is where we turn our attention to our 2 key lipases that we've discussed in previous lectures. Hormone-sensitive lipase is one of them. Remember, hormone-sensitive lipase plays a role in liberating free fatty acids from triglycerides so that we can use those free fatty acids for fuel. What the study looked at was hormone-sensitive lipase mRNA expression as well as protein content. What they found was that females have greater mRNA and protein content for hormone-sensitive lipase. So women in the black bars, men in the white bars, females are higher regardless of rest or during 90 minutes of exercise. Importantly, what we touched on in class is this idea that we wouldn't expect to see changes in mRNA or specifically protein content in such a short time frame. Changes in protein content typically occur over 24 to 48 to 72 hours, so hours to days timescale, not within an hour and a half. But really, when we think about what's missing from this picture, changes in hormone-sensitive lipase mRNA content or protein content is great to know, but we need to really think about hormone-sensitive lipase activity. So when we consider protein activity, we know that we can use phosphorylation, dephosphorylation as a means to activate or deactivate an enzyme. So in this case, just a reminder or a heads up that phosphorylation of hormone-sensitive lipase, in this case on serine 659, results in the activation of hormone-sensitive lipase. So what they did here was on the y-axis, you'll see that they expressed phosphorylation per total hormone-sensitive lipase protein content. So this is super important because we know from the previous slide that females have greater hormone-sensitive lipase protein content, so it wouldn't be fair to express the data or the phosphorylation of hormone-sensitive lipase in absolute terms, because we'd expect with more protein content you would have more phosphorylation. So this way we can express it relative to each hormone-sensitive lipase protein, how much of them are actually phosphorylated. What we see is that with exercise, so the cross there, it signifies that exercise increases hormone-sensitive lipase activation. Makes sense. When we start our exercise bout, we need to liberate free fatty acids for fuel, so hormone-sensitive lipase is going to play that role. What we also see with the astraces is that males have greater hormone-sensitive lipase phosphorylation at rest and following 90 minutes of exercise compared to women. This might seem interesting, but what we need to consider is that there's multiple residues on hormone-sensitive lipase, and phosphorylation of one doesn't always translate to changes in activity. So let's kind of go forward, look at hormone-sensitive lipase protein activity, and see what we find. When we look at hormone-sensitive lipase protein activity, what we see, again, we express hormone-sensitive lipase activity relative to the total hormone-sensitive lipase protein content. Again, because females have higher protein content, we want to express it relative to each HSL protein itself. What we see, again, exercise with a cross increases HSL activity. Makes sense. We need to liberate those free fatty acids for fuel. What we see is the same trend again, in that males have greater hormone-sensitive lipase phosphorylation, but they also have greater hormone-sensitive lipase activity at rest and following exercise compared to females. So you guys are probably having a bit of confusion here, so you're probably thinking that doesn't really make sense. We want to see females have greater ability to utilize fat, but this is an interesting finding where again, it's opposite compared to where females have higher hormone-sensitive lipase protein content, males seem to have more activity. So when we take this together, hopefully our brains don't explode, but really this idea that females have this higher protein content at rest and following exercise, males have this greater phosphorylation and activity. So when we express it in absolute terms, what we see is that there's actually no differences between men and women with regards to hormone-sensitive lipase, phosphorylation, and activity. Remember, this is in absolute terms. Why that makes sense is because females have more protein, but it's less active. So they're dispersing the workload over more proteins, but they're not doing as much work. Whereas males have less protein, but they're doing more work. So in the end, it's balancing out that there's no difference in the activity between men and women. It's just how those differences in activity are kind of brought about. But missing pieces of this puzzle that have yet to be investigated. We know ATGL and MGL are important lipases for liberating free fatty acids from triglycerides. So maybe these are avenues that future research can consider. Maybe sex differences reside here, especially because we know ATGL is considered this rate-limiting step of lipolysis. So the last kind of thing I want to touch on with lipid metabolism and potential sex differences is this idea of mitochondrial lipid transport. So we know that we need to bring that free fatty acid across those mitochondrial membranes in order to undergo beta oxidation, Krebs cycle to electron transport chain. And we know that CPT1, or carnitine palmitoyl transferase 1, is the rate limiting step for fatty acid transport into the mitochondria. What we see looking at the bar graph here is that we're looking at CPT1 mRNA expression. We see that women have higher CPT1 mRNA expression, but when we move over to the linear regression figure, we see that this doesn't again translate to one protein content, which isn't shown, but this figure is showing us CPT1 activity. So what we actually see is that there's a relationship between fitness level, so VO2 max on the x-axis and CPT1 activity on the y. No sex difference, but the more fit an individual is, the higher their CPT1 activity. If they're endurance trained, this makes sense. So VO2 max is an aerobic capacity test or an aerobic endurance test. Greater VO2 max, greater endurance capacity, greater reliance on fat for fuel during exercise. Makes sense, we might see greater CPT1 activity. But again, it doesn't translate to a sex difference in mitochondrial lipid transport. So that's a lot of information. Taking it all together, what we can see and what we can say is that females rely more on fat for fuel during submaximal exercise. They have greater fat stores, as well as intramuscular triglyceride stores, greater ability to utilize those intramuscular triglyceride stores during submaximal exercise. But the literature isn't quite there yet to determine how we actually use those lipids to a greater extent, since there's no differences in HSL, there's no differences in CPT1. But again, we've left some avenues that are yet to be explored to explain the differences in lipid metabolism and how females actually utilize a greater proportion of fat relative to males. Now we're going to shift gears, talk about sex differences in carbohydrate metabolism. So again, red box, ignoring lipids this time, switching gears, looking at carbohydrate side of things, thinking about glucose in the circulation, bringing that into skeletal muscle or intramuscular glycogen stores, how we break those down to eventually form pyruvate, which can undergo aerobic metabolism in this example, or through lactate dehydrogenase, we can have anaerobic glycolysis as well. So first study I want to point out is looking at glycogen metabolism. So 19 males, 28 females. What we're thinking about here is a 15.5 kilometer treadmill run at a moderate intensity, so 65% of their VO2 max. What they looked at was muscle glycogen concentrations as well as plasma glucose concentrations during the exercise bout. So the glycogen they measured was pre-post exercise in men and women. So top figure there. What we see is that following exercise, females use significantly less glycogen for a submaximal endurance task. So what we're looking at here is the white bars is female, black bars is males. Females use less muscle glycogen to complete that task. Furthermore, what we see is that the black circles is males on the bottom figure and the white open circles is females. Females have significantly less glucose uptake into skeletal muscle. So what we're looking at here is this idea that this is likely due to less liver glucose metabolism. So it appears women rely less on muscle glycogen and less on glucose metabolism for fuel provision during exercise. But what other evidence do we have to support this? So we can actually look at the enzyme activities of a variety of key enzymes in carbohydrate metabolism. So first, GP is glycogen phosphorylase. Remember, glycogen phosphorylase commits glycogen to be broken down into glucose and that glucose to form glucose 6-phosphate or G6P. What we find is that glycogen phosphorylase activity is significantly lower in females, so the gray bars compared to males in the black bars, suggesting a reduced ability to commit glycogen to breakdown and energy provision through glycolytic pathways. What we see when we think of another key enzyme, hexokinase, similar story. So what hexokinase does is actually commit circulating glucose to be brought into the skeletal muscle and actually converted into glucose 6-phosphate so that it's being held within that skeletal muscle. Same story, gray bars is women, black bars is males. Women have lower maximal hexokinase activity compared to men, suggesting that women again have this reduced ability to commit glucose to staying within skeletal muscle for energy provision through glucose metabolism pathways. Lastly, thinking about PFK or phosphofructokinase. Remember, PFK is the committing step for glucose oxidation. So what happens is that PFK actually turns fructose 6-phosphate into fructose 1,6-bisphosphate. What we see again, gray bars female, black bars male. Women have lower maximal PFK activity compared to men. Not surprisingly, same story. Women have a reduced ability to commit glucose to energy provision through these glucose metabolism pathways. So big picture, this is all from the same study. What we're looking at is glycolytic enzyme activity, specifically maximal glycolytic enzyme activity. Taken together, big picture, females appear to have reduced maximal glycolytic enzyme activity compared to men. But as discussed in class, we know maximal enzyme activity is a problem in the context of true physiological differences or physiological scenarios. So what we need to consider is submaximal flux. So this study by Ropestorff et al., 2006, looked at acute glycolytic regulation during submaximal exercise. So 90 minutes of submaximal exercise, moderate intensity. What they looked at was epinephrine and norepinephrine levels, epinephrine and AMP and ADP levels during exercise in men and women. The reason why they did this is that because maximal enzyme activity doesn't necessarily represent the conditions experienced during exercise, and we know that glycogen phosphorylase activity is fine-tuned by metabolites, so ADP, AMP, as well as catecholamines, example, epinephrine. So looking at the figures on this slide, what we see is that plasma epinephrine levels are significantly higher in men compared to women. Men are in the open squares, women are in the closed circles. Again, plasma epinephrine levels significantly higher in men compared to women during a bout of submaximal exercise. Moreover, free AMP significantly higher at the end of that submaximal exercise bout in men compared to women. Men in those open squares, women in the filled circles. Remember, glycogen phosphorylase, so breaking down glycogen to glucose, committing it to glucose 6-phosphate, subsequently oxidizing it for fuel. Glycogen phosphorylase activity is fine-tuned by metabolites and catecholamines, meaning that higher levels of AMP, ADP, epinephrine can all increase or promote glycogen phosphorylase activity. This suggests another way that females may have a decreased reliance on glycolytic metabolism compared to men, because they have lower epinephrine, lower AMP, ADP in response to the same exercise challenge. They may have less activation of that glycogen phosphorylase, less breakdown of glycogen for fuel provision. So bringing it all together, fat, lipid, or carbohydrate metabolism, what we see is that women rely more heavily on intramuscular triglycerides and plasma-free fatty acids, so lipids, whereas men have a greater reliance on carbohydrates for fuel during the same exercise challenge at 50% VO2 max. Big picture again, females rely more on fat, males rely more on carbohydrates. Now I want to take some time to shift gears and look at some post-exercise measurements before we talk about protein sex differences or sex differences in protein metabolism. So thinking about glycogen resynthesis in the post-exercise period, what the study did was recruited 8 males, 8 females, exercised submaximally, so moderate intensity for 90 minutes. They then took biopsies immediately post-exercise and 4 hours post-exercise to look at rates of glycogen resynthesis. Participants were actually given carbohydrates immediately after the first biopsy, as well as 1 hour post-exercise to augment those glycogen resynthesis rates. What they found Absolutely no difference between men and women in their capacity to resynthesize glycogen in recovery. However, when we turn our kind of focus to carbohydrate loading, a common technique used by athletes in order to enhance their exercise performance, this study looked at if there were sex differences in the capacity to carbohydrate load. 7 males, 8 females. They put them on both, so 2 different kind of test scenarios. A low carbohydrate diet for 5 days. They then switched over and did a high carbohydrate for 5 days. In the high carbohydrate diet, it was matched between men and women that 75% of their caloric intake was going to be carbohydrates. That's a pretty big proportion of a daily diet and is going to be considered what's normal if we're going to have a high carbohydrate diet. What they did was they took blood samples, muscle biopsies. What their actual exercise bout was, though, was 75% of VO2 peak for an hour. They took a rest period. They then performed 85% VO2 peak to exhaustion. So both of these exercise intensities are purposefully high intensity because we think and we remember back to our figure at the beginning of the lecture. High intensity exercise, we rely on carb for fuel. This is how we're going to drive and tease apart differences in how substrates are utilized between men and women in response to carbohydrate loading. What did they find? High carbohydrate diet versus low carbohydrate diet. Men had a significant increase in that 85% VO2 peak to exhaustion. They had a 45% increase in performance. So 45% increase in the time to exhaustion at 85% of their VO2 peak. Women, on the other hand, a small 5% increase in performance, and it was non-significant. What I want to highlight here, a typical carbohydrate loading protocol is about greater than 8.0 grams of carbohydrate per kilogram body mass per day. You guys can do the math, what that means for you. But now we need to think about why there's a sex difference here. What's actually happening? So when they looked at their muscle biopsies, one of their key measures was obviously muscle glycogen concentrations. What they found was that in response to the high carbohydrate diet, men, so in that first red box there, from low to high carbohydrate diet, 401.5 to 565.4, significantly more muscle glycogen in males following that high carbohydrate diet. They successfully carbohydrate loaded. Females, though, if we take the low carbohydrate diet in that second box, 407.3 compared to 407.8, absolutely no change in muscle glycogen in these women following a high carbohydrate diet. Really emphasizing this idea that they're unable to carbohydrate load. And what we see is that the reason behind this, and really the reason why carbohydrate loading does not work in women, is this idea that we're coming back to this 8 grams of carbohydrate per kilogram body mass per day. If women consume more than 8.0 grams of carbohydrate per kilogram body mass per day, in the context of a 2,000 calorie diet or however many calories they should be consuming, this would be 93 to 120% of their daily intake, strictly coming from carbohydrates. Obviously, 100% of your diet is carbohydrates. That sounds awesome, but it's not feasible. We need proteins, we need fats in our diet in order to obviously have proper health, proper physiological function, proper metabolic function. So it's simply not possible for women to consume this much carbohydrate in the context of a normal calorie diet. Of course, they could consume this much carbohydrate in the context of overeating, so over-consuming the calories needed for their body mass. But this comes with other negative implications for athletes that would want to do this, because you would have weight gain, which is obviously, in most scenarios for top tier athletes, not a beneficial effect or not something you want to accompany your training protocol. So now we're going to shift gears one more time, think about sex differences in protein metabolism. Recall, so we've had a review on carbohydrate, had a review on lipids. This is our recall for protein metabolism. Protein turnover is comprised of synthesis and breakdown. So synthesis being taking amino acids, making new proteins. Breakdown being taking whole proteins, chopping them up into individual peptides, taking those peptides, breaking them down further into amino acids, where they can then continue to cycle again back to synthesis of other proteins. Some of those amino acids might also go towards oxidation, or in other words, metabolism. But really, when we consider protein balance, it's this idea of the ratio of protein synthesis to protein breakdown, where if we're in an anabolic state, synthesis is exceeding breakdown. In a catabolic state, breakdown's exceeding synthesis. So when we look at resting protein synthesis, so if we're just hanging out in the classroom, what is the difference between men and women in terms of protein synthesis rates? Remember, fractional synthetic rate is the same as muscle protein synthesis. Men, it's originally hypothesized that men have greater muscle rates of protein turnover, so more synthesis, but also more breakdown, aka more turnover. But really, this might be a product of greater lean mass in males. So what these authors actually did was they expressed their data relative to lean body mass. So they normalized their data to lean body mass, and they found that muscle protein synthesis had no significant difference between males and females. It's important that these authors normalize their data to lean body mass because, of course, when we're talking about protein synthesis, protein turnover, lean mass is of interest because skeletal muscle mass will differ between the sexes. When we talk about protein synthesis in recovery, so what they actually did was they had 14 males, 16 females. They did an hour of resistance exercise, one-legged resistance exercise. They compared the FP, so follicular phase, and the LP, luteal phase, for menstrual cycle differences, and that's that bar graph on the side there. What they found was that there's no difference between the luteal phase and the follicular phase at rest or 24 hours in recovery for protein synthesis rates, so menstrual cycle does not play a role here. And they also found, looking at the table on the bottom there, no sex differences between men and women in terms of protein synthesis or fractional synthetic rate 24 hours post-exercise. So again, no sex difference, no menstrual cycle difference with regards to protein synthesis in recovery. So what these authors, another group of authors sought to investigate a similar question. They wanted to look at post-exercise hormonal responses. So what they looked at here was this idea that testosterone is typically thought to be linked to protein synthesis, more lean mass, more gains kind of thing. What they looked at was this idea that it's thought that testosterone levels are elevated following a resistance exercise bout, and that this might augment or shift muscle protein synthesis. So looking at panel A here, we see testosterone levels in the men in the black dots increases during and after that exercise bout compared to baseline, returns to baseline at 24 hours. In women, the white circles, no change in testosterone levels. So men have a greater area under the curve for testosterone, obviously, as they have higher baseline levels to begin with, and they see this peak during exercise as well. The other thing to consider is that males are able to convert testosterone to estrogen. So these elevated testosterone levels actually might be able to go towards estrogen as well. And what they see is that men in the black dots on the estradiol figure here and women in the white circles, women's levels of estrogen do not change over time in response to exercise or recovery from exercise. But in men, we see the same thing where during exercise, there's a small rise in estradiol levels that's maintained at 24 hours into recovery. So not only do men have these higher circulating testosterone levels, but they also have this rise and sustained rise in estradiol levels. These authors thought that this might contribute to a difference in protein synthetic rates between men and women, and actually how muscle protein synthesis occurs. But what they found that after that single bout of resistance exercise, and they studied them 5 hours into recovery, those hormonal status markers, whether it's testosterone or estrogen, had no real influence on post-exercise muscle protein synthesis when measured from 1 to 5 hours into recovery. So it's kind of washed out their hypothesis, but really again showed that the results match between this study and the previous study, where there's no sex difference in muscle protein synthesis between men and women in recovery from exercise. And the last slide I want to talk about is a training study, so resistance training. Large cohorts here of males and females, 12 weeks of resistance training. Big take-home picture was that males had a greater increase in muscle cross-sectional area, so muscle size, but females had a greater relative increase in 1 rep max, or the greater increase in strength compared to males. Taken together, this suggests that resistance training may result in greater gains in muscle size, muscle cross-sectional areas in males, but females may have a more functional outcome as a greater relative increase in muscle strength compared to males. So with that ends the lecture content for the midterm. Remember that Thursday's protein presentations are also testable content for the midterm. And next week, you guys don't have to come in. It's reading week. Take this time to study, prepare for the midterm. Again, midterm, February 27th, in class, 9:30 to 11. In preparation for the midterm, please think of some questions for over reading week for our review lecture on Monday after reading week.